Hello friends, this is Dr. Archana Kulkarni and welcome to my video on Methy Red Test. Friends, Methy Red Test is the second biochemical test in the consortium of IMViC Tests and is used for the identification and differentiation of bacterial species. This test is used to determine the ability of bacteria, primarily enterics, to oxidize glucose using mixed acid fermentative pathway to produce stable acid end products which will overcome the buffering capacity of the medium used. The product of this mixed acid fermentation are a mixture of acids, particularly lactate, acetate, formate, succinate, as well as ethanol and equal amounts of CO2 and H2. Friends, due to the accumulation of these acidic products in the medium, the pH of the broth changes from pH 6.9 to pH 4.4 or less. So when the pH indicator dye like methyl red is added to a test culture, it produces a deep red color indicating mixed acid fermentation, and the test is regarded as MR positive. And the example is E. coli. Friends, but other enterics like Enterobacter aerogens, Klebsiella pneumoniae, however, follow 2,3-butanediol pathway to oxidize glucose, and they produce neutral end products like acetyl-methylcarbinol, which is also called as acetoin, then diacetyl and 2,3-butanediol. So when methyl red indicator is added to a test culture, these bacteria will produce yellow color, indicating that the MR test is negative. This is because here the broth pH remains either 6.2 or above, and that is why the MR test is negative. Now let us see the characteristics of methyl red indicator. Friends, methyl red is a red crystalline powder. It is a pH indicator. This methyl red is acid sensitive azo dye. It is yellow at pH 6.2 and above and red at pH 4.4 or below. Here in this diagram, the structure of methyl red is shown, and based on the pH variation, different colors that it gives are shown. Now let us see the requirements of methyl red test. To perform this test, you require glucose phosphate broth. This broth is commercially also known as MRVP medium, or it is also called as buffered peptone glucose broth. Then you will require a fresh culture of 24-hour-old test bacteria and a reagent, that is methyl red reagent. Now we shall see the composition of media and reagents. Friends, this glucose phosphate broth contains buffered peptone 7 grams per liter, glucose 5 grams per liter, dipotassium phosphate 5 grams per liter, distilled water 1000 ml, and pH is set to 6.9 plus minus 0.2, that is pH is neutral. Now we shall see the composition of methyl red reagent. To prepare 100 ml of this reagent, you will have to weigh methyl red 0.200 grams, ethyl alcohol 60 ml, and distilled water 40 ml. Friends, now let us see the procedure. First, you prepare a glucose phosphate broth and distribute 5 to 10 ml of it in the rimless glass test tubes. Sterilize these test tubes in an autoclave at 15 pound pressure at 121 degrees Celsius temperature for 15 minutes. After the sterilization is over, inoculate the broth tubes aseptically with the given fresh test culture and incubate at 37 degrees Celsius for 18 to 24 hours. After the incubation, you add 5 drops of methyl red reagent to the broth culture and observe for the presence or absence of deep red color. The presence of deep red color shows that sufficient acid has been produced in the broth to lower the pH to 4.4 and the MR test is positive. If the color remains yellow, that means MR test is negative. Here in this photograph, the positive and negative MR tests are shown. Now we shall see which are the organisms that give MR test positive. Friends, bacteria like E. coli, Proteus vulgaris and Salmonella typhi gives MR test positive. And bacteria like Enterobacter erogens, Klebsiella pneumoniae gives MR test negative. Now we shall see the biochemistry of Methy Red test. Friends, the Methy Red Voges-Proska tests were originally used to distinguish between members of the family Enterobacteriaceae, but now are also used to characterize other groups of bacteria, including actinobacteria. Enteric bacteria like E. coli and Enterobacter erogens are facultatively anaerobes and use aerobic respiration in the presence of oxygen. And anaerobic respiration if alternate electron acceptors like nitrate, fumarate, sulfate, etc. are available. In absence of any such electron acceptor, this bacteria will enter into the fermentative pathway. And two such fermentative pathways found in this bacteria are mixed acid fermentative pathway and the second one is butylene glycol or 2,3-butanediol pathway. The relative amounts of each product produced during fermentations here may vary with growth conditions and bacterial species involved. Friends, in E. coli, it follows mixed acid fermentative pathway. And this mixed acid fermentation occurs in two stages. In the first stage, glucose from glucose phosphate broth is anaerobically oxidized to pyruvate, which is the end product of this glycolytic pathway. And further, in the second stage, this pyruvate is converted to one or more end products using different pathways which requires different key enzymes. And after the different end products are formed, they are secreted from the cell. So, lactate is produced by reduction of pyruvate using the enzyme lactate dehydrogenase. Acetate is produced when pyruvate is first converted to acetyl-CoA and formate. In the presence of enzyme, pyruvate formate lies. The acetyl-CoA is then converted to acetate by the activity of two different enzymes, namely phosphotransacetylase and acetate kinase. And the formic acid breaks down into the gases like CO2 and H2 in the presence of enzyme formate hydrogen lyase, which is very typical in E. coli. Ethanol formation is established using the enzyme alcohol dehydrogenase, which catalyzes the reaction from acetyl-CoA to ethanol. Succinate formation starts with the carboxylation of phosphoenol pyruvate to oxaloacetate by the enzyme phosphoenol pyruvate carboxylase, and is subsequently achieved via the activity of three different enzymes, namely malate dehydrogenase, fumarase and fumarate reductase. So this is how different acids are produced, and D-glucose is completely oxidized into number of acids like lactic acid, acetic acid, formic acid, succinic acid, ethanol, CO2 and H2 as shown in the pathway. Friends, mixed acid fermentative pathway gives 4 moles of acidic products, 1 mole of ethanol and 1 mole of CO2 and H2 each per molecule of glucose.